Hello, everyone, and welcome. Thank you all for joining us today. I'm Julien Gillet-Daubin. I'm the Director of Capital Cell for France, and it's a pleasure to have you with us for this webinar. Today, we will focus on the evolving landscape of the IBD and a new therapeutic solution based on siRNA with Oragen, who will present us their technology and their ambitions. Our session today will include about 5 minutes of introduction of a different person you can see on the screen, followed by 20 minutes of pitch, and then 5 minutes for Jean-Marc, who will present ARA, Health Angel Rhône-Alpes vision about their investment on Oragen, and then we will have 20 more minutes for Q&A. And feel free to submit your question writing via the Q&A box that is just below the video screen. So without further ado, let's get started with a presentation of the panelists that we have with us tonight. So maybe Henri, I'll let you introduce yourself.
Thank you, Julien. Good evening, everyone. I'm excited to have the opportunity to introduce you to our ambitions. So I'm a former pharma executive, Doctor of Pharmacy by training, and the CEO and co-founder of Oragen Therapeutics. And I will hand over to Arnaud.
Hello, everybody. Arnaud Béduneau. I am the CSO and co-founder of Oragen Therapeutics and inventor of the patent exploited by Oragen.
Jean-Marc.
Yeah, hello. So I'm Jean-Marc Renard. I'm co-secretary of ARA, which is Health Angels Rhône-Alpes. And in my previous life, I was working for Sanofi Pasteur, and I was vice president corporate development, which covers business developments and M&A functions.
Thank you very much. Henri, I leave you the floor for your presentation.
So let's get started. Can you see my screen?
Yeah.
Great. So we believe at Oragen Therapeutics that breakthrough innovation starts by asking a bold iconoclastic question, a kind of what-if question. And in our case, this is the following. What if we could combine the potency of genetic medicines with the simplicity of oral administration? And spoiler alert, we can. We have demonstrated that in animal models. And this is transformative in a number of applications, starting with the so-called IBD, Inflammatory Bowel Disease, Crohn's disease, and ulcerative colitis. These are really devastating diseases, immunoinflammatory disorders associated with pain, fatigue, and a strong disability of the patients suffering from those diseases. And there are over 10 million patients affected by IBD worldwide, with 6 million uniquely in Europe and North America. Of course, the healthcare costs associated with those diseases are extremely high because they represented over 30 billion annually in the US and Europe. And if we dig further into this market from an economical point of view, this is a growing market, which was over 20 billion on an annual basis last year. Well, 2 years ago now, in 2024. And this market is expected to grow beyond 33 billion by 2032, driven mainly by the increase of the prevalence of this disease, which is expected to grow in US and Europe from 6 million patients to beyond 8 million by 2030. So there is a huge unmet need and a huge market by size. And this market, apologize for this busy slide. Let's focus on 3 points on the current landscape of the therapies approved in IBD. First, one thing to notice, 2/3 of the treatments approved and used in the market are monoclonal antibodies, and these are strong implications from a scientific point of view. Let's also focus on one thing. All drugs approved in IBD generate over $1 billion on an annual basis. And the last point to notice here is that almost all those drugs will lose their market exclusivity by 2032, which means from a strategic point of view that it drives a strong need for all the actors, the players on the IBD market. And you can see these are large pharmaceutical companies. They need to refuel their pipeline with new assets developed by biotech. And what asset to develop? The assets that will fulfill the unmet medical need in IBD. And there is a huge unmet need. Because you know what? Up to 80% of patients suffering from IBD fail to achieve sustained remission with the current therapies, especially for Crohn's disease. And for a significant portion of these patients, this is due to the development of ADA. ADA stands for anti-drug antibodies. It means that the body produces antibodies against the monoclonal therapies that they use as a treatment. On the other side, there are huge safety hurdles currently because the treatments on the market, most of them induce rare but severe side effects. We are talking about risk of cancers, of infections. And beyond all, what do IBD patients want? They want oral treatment, whereas most of the drugs approved are injectable. So this is our answer to this need. Using the potential and the potency of genetic medicines, and namely RNA, to fulfill this unmet need with our breakthrough approach. This approach is a 3-layer structure. This is our technology. That's a platform technology that enables the development of several drug assets. This technology encapsulates first, at the core, a nucleic acid payload, RNA, therapeutics here. It's loaded within lipid nanoparticles, which basically is the kind of structure that we had in the COVID vaccine or the drugs currently approved on Patro. The core of the novelty lies in the proprietary microparticles that has 2 key properties. The first one is gastroresistance. So we make RNA resistant to pH and to enzymes. And secondly, we have the capacity to address them directly to the inflamed areas of the bowel. So these 2 key properties are really what makes our technology different from the others: gastroresistance and targeting. And let's dig further into the science with you, Arno. Oh, no. Before moving to the science, let's try to make an analogy. Basically, for those of you who think, what is our technology? That's a kind of armored ambulance with a GPS. We protect and we target the RNA where it matters. The floor is yours, Arno.
Okay, thank you. So our macroparticles specifically target the inflammatory lesions in the intestine due to their size and the surface charge. As shown in the left diagram, the size of macroparticles, the macrometric size, prevents them from penetrating the healthy mucus layer on the surface of the intestinal mucosa. In contrast, so it's on the right, in contrast, this mucus barrier is altered at a site of inflammation, allowing macroparticle adhesion and the specific release of the payload directly in the inflammation lesions. In addition, the negative charge of the polymer forming the macroparticles can interact with the cationic peptide located at the inflammation site. So we have a kind of electrosteric targeting of inflammation lesion. Inflammatory
That's a good rationale, Arno, but have we demonstrated that?
Here, we administer our macroparticles labeled with a fluorescent probe orally in a murine model of colitis, but also in healthy mice. And after that, we collected the different segments of the intestine and we measure, we quantify the macroparticles in different intestinal segments. And we observe the highest concentration is in macroparticles is located in the inflamed colonic region. In contrast, the healthy segments of the intestine, both in healthy mice but also in colitis group, we have a very low concentration in macroparticles. So these results demonstrate clearly the specific targeting of inflamed intestinal lesion, but also the preservation of healthy segments. Here, this is the evaluation in this diagram. This is the evaluation of the efficacy of ORA-251 in a TNBS-induced murine model of colitis. After a daily oral treatment for 3 consecutive days, the therapeutic index of treated mice, so this is the green bar, and the therapeutic index is defined as the ratio between the colon weight and the length. So this therapeutic index was significantly reduced compared to untreated mice, so dark blue bar. And this therapeutic index was even comparable to that of the healthy mice. So this is the light blue bar, demonstrating the complete remission of the animals.
That's really the key point to your closure. Close to remission after 3 days of treatment. And we compare that to other drug being either on the market or in development, and that's striking. And so you understood, so that's a bit of teasing. ORA-251 is a lead asset, and that's part of strategy. Why is this interesting as a drug asset? Because it plans for improved efficacy based on preclinical data, but also based on the modality. This is the first oral siRNA to treat inflammatory bowel disease. And this drug modality is not subject to anti-drug antibodies, so no wane of efficacy over the time, which is one of the limitations of current therapies. We have demonstrated its gut-restricted approach, which is very good indicator for a good safety profile. And beside all this, it's an oral treatment where the competitors are injectable. So that's, of course, better for the patient adherence to treatment. So we have a differentiated technology and we have a differentiated asset. How to make the most of it in terms of value creation? Our strategy relies on 3 pillars. First, generate clinical evidence. That's an important aspect to this platform technology to validate its applicability in the clinic, so in human. And we plan to start our first human trial within the next 3 years with ORA-251 that will exemplify the potential of our technology. Second, we plan to demonstrate the versatility, meaning the capacity of the technology to address different diseases or to load different active ingredients, different siRNA. And we plan to conduct within the next 12 months additional studies in colorectal cancer, notably. And third, we operate a de-risk model. De-risk why? Because we have a dual value creation engine combining both assets and platform. We develop a clinical asset that has value, and a strong one you will see based on the analogs. And secondly, we have a technology that can be out-licensed in non-strategic indications to other pharma companies. Besides, from a biological point of view, we don't reinvent the so-called target, the biology. We leverage mode of actions that have been demonstrated to work, but we amplify their potential with our technology based on RNA. From a development point of view, we plan gradual funding to reach desired value inflection points. The first one here, which is the subject of this current pre-seed round, is to reach a stage which is so-called ready for IND, which means that we are ready to start the CMC and GLP toxicology studies preliminary to the clinical trials. We plan also to reach for this first value inflection point after this pre-seed round to demonstrate the versatility of our platform, notably in the other indications I mentioned, colorectal cancer, but also by loading several siRNA, which is something that generates significant traction among the key pharmaceutical players. For this, we are planning a 2 to 2.5 million euro overall funding, non-dilutive plus dilutive. And we can see the breakdown after. The second stage is to reach the clinical stage with a lead candidate that will demonstrate in the clinics the potential of our technology. For this, we are already in touch with dedicated venture capitalists in France, in Europe, mostly, that will fund this stage, this 40 million stage that will help us move to the clinic and then even beyond. So that after stage 2, from 2029, we have generated clinical evidence of our technology. We have demonstrated the versatility of our technology. So we are a pretty interesting target for M&A activities. And that's not an internal conviction. This is based also on the interaction that we currently have with 6 out of the 10 key pharma players, global pharma players currently. Depending on the traction and on the market condition at that time, either we sign a deal or we can continue the development to move up to phase 2, which will further increase the valuation of the company. Beside, or not beside, but this is our core engine, I would say, the team behind Oragen Therapeutics. The executive team made of the co-founders as well as a pharma veteran experience in drug development, Didier Cussac, with experience at Servier Pierre Fabre in terms of really R&D within pharmaceutical companies. I'm the CEO of the company. I have 15 years of experience in the pharma business, spanning large pharmaceutical organizations, namely Sanofi, Merck joint ventures, as well as more recently a biotech company called Valneva. I oversee the strategy of the company and have experience in strategy, but also in business development and in market access, which is important, I think, to drive a biotech company. Arnaud, who is our CSO, is really experienced in terms of drug delivery and inflammatory processes, as well as Dr. Thomas Talder, who is a hospital pharmacist and skilled in drug delivery and analytical chemistry. This is the executive team. Besides, we are advised by 2 world-leading boards. We have in our scientific advisory board 3 worldwide experts, Professor Laurent Perrin-Piroulet, which is probably the #1 key opinion leader in the world in Crohn's disease, and who have made the choice to join this venture with us. We are grateful to have someone skilled like him. We are also advised by Professor Lucine Vitton, which is a European-level key opinion leader, as well as Professor Alexander Kabanov, who is really a worldwide US-based but worldwide expert in nanomedicine. Beside this, we have 2 strategic advisors, pharma biotech veterans, Pierre Morgon and Philippe Archinard, who provide us with a strategic guidance, which is extremely important, as well as networking opportunities among the pharmaceutical community, I would say. We are currently seeking for a 2 million pre-seed funding, including 6.6 million euro equity. How are we planning to use this proceed? First, to generate this clinical evidence. What do we mean by that? Finalize the construction of a proprietary small interfering RNA. Conduct the necessary study to confirm its effective dose, as well as its preliminary toxicology assessment. So then we have the risk, the efficacy, and the toxicology. Secondly, demonstrate the versatility of the platform and start right away to prepare the industrialization of our technology with more precisely the microparticle, which is the core of the novelty, because the rest of the technology is already widely used in the industry. And we have already discussion with CDMOs to confirming that there is no blocking point on this, but it needs to be formalized under specific manufacturing processes. And then establish the operational foundation of the company in terms of recruitment and process implementation, including quality, finance, and I would say the standard G&A operations. Why are we attractive? So beyond developing a first oral drug candidate, which will be a real breakthrough in the biopharma world, we are working on something that's extremely attractive for investors. Because what? Look at the data. The mean valuation of inflammatory bowel disease drug candidates, not on the market, but drug under development. Over the last 5 years, there have been 8 deals with a mean valuation of 3.1 billion. So that's really a huge market opportunity for drug candidates. And that generates also significant traction among the key pharma players because there is huge market potential, a huge unmet need, and assets that demonstrate signals of efficacy and proof of safety, as we plan to do, can, I would say, hope for over $1 billion valuation. And beside, we have also valuation of the platform technology, and we can dig further into that later on during this discussion. So why us? Because we can make oral RNA therapy a reality. We have a revolutionary technology with a differentiated drug candidate, and that's an effective and versatile platform for oral delivery of nucleic acid with a first-in-class oral siRNA and applicability beyond inflammatory bowel disease. We have already reached significant milestones. I strongly believe that the momentum of the company is one of these key assets. Generating compelling preclinical data already, we have been awarded as one of the most promising startups by BioEurope in 2025, and we have established world-class advisory boards. And maybe that's the key to conclude. We represent a massive opportunity because there is a huge unmet need in IBD, well informed in the business. That's a commercial opportunity over 30 billion, and the deal analogs for the drug under development are over 1 billion. We are already supported by a strong network of ecosystem, and we are glad to discuss with you to get your support as well.
Thank you. Thank you very much. Now, what I would propose is maybe Jean-Marc, we give you the opportunity to present the point of view from ARA, and maybe a quick refresh about who is ARA, and then maybe the reason what you liked and what you fear maybe also in this project. But what makes you making today an investment on this project?
So ARA is an association of health angels, and we have about around 40 members. And when we decide to go into projects such as and invest into projects such as Oragen, we usually do it on an individual basis. So it's not like ARA invest and everybody has a share of ARA's portfolio. It's each individual who decides to invest in a company. So we have thoroughly reviewed Oragen together with Capital Cell, And the reason why we find this opportunity exciting and we'll invest is because it's from our point of view, it kind of checks all the boxes that we want to see into innovative research projects. It addresses a large market, so we know that if the product makes it to the market, there is a large commercial opportunity. But at the same time, it's also a market where the unmet medical need is still high. So that's a winning combination. Large markets with unmet medical need means that there is still some room for new products, for improved therapies. And what we also find particularly attractive is that it builds upon a well-established and demonstrated mechanism of action. Anti-TNF alpha antibodies are the first-line treatments for IBDs. So we know it works, and we know that by reducing the amount of TNF alphas, you actually address the disease. But by the same token, what is also very interesting with Oragen's approach is that it addresses the weak points of the existing therapies, which, as was explained by Henri, is the appearance of anti-drug antibodies. And those anti-drug antibodies, if my memory is correct, as a result, mean that more than 40% of those patients who at first respond to anti-TNF therapies become resistant to those therapies. And what is very elegant in the approach from Oragen is that by using a new mechanism, which is instead of reducing the amount of circulating TNF alpha by antibodies, it reduces the circulation of TNF alpha by reducing the synthesis. So by having siRNA delivered to the cells, which usually produce TNF alpha, you actually do reduce the TNF alpha concentration. And then the beauty is that given that TNF alpha is reduced, then there is no room for anti-drug antibodies to develop. So if the product delivers, if the approach delivers, then it means that we have a therapy that will be a big improvement over the existing anti-TNF alpha approaches. And what we then find very attractive also is that Oragen has been able, although it's a relatively young company, they have been able to demonstrate not only in vitro, but also in vivo and in animal models that their approach does work. And that's impressive because it's not a given. As was explained during the presentation, in order for the product to work, you have to have the microparticles actually go through the GI tract without being destroyed. You have to have them delivered at the place where they need to be, and then you have to have the siRNA to be delivered and to work and to reduce the TNF alpha concentration. So all of those are hurdles which really are not obvious. And I think it is impressive that Oragen has been able to demonstrate that into the TNBS induced colitis model, which is the model of reference for colitis. So again, for us, that means that the mechanism of action, it builds upon a demonstrated mechanism of action with an approach which has been demonstrated to work in vivo in animal models. So that means that the probability of success from a scientific and technical point of view seems relatively high. So again, we're combining a big market, a big unmet medical need, with an elegant approach which addresses the weaknesses of the existing therapies with what we feel has a good probability of success. So all of this combined with a team which seems to be of good quality and a development plan which seems to be of good quality makes it, from our point of view, an attractive investment opportunity.
Thank you. I think it's quite clear. I just take the opportunity before going to the Q&A session that we recently published on the blog of Capital Cell an article where we try to explain for those of you who are probably not always experts in difference between RNA, mRNA, siRNA, and everything. So I think it was last Sunday we posted an article explaining all those things so beginners can have a better understanding on their different way and mechanism of action for that. Well, we've got already a few questions. So I take them directly in order. Regarding the technology, could your carrier have any other kind of drugs than a nucleic acid? Could you have something else within your carrier? And if yes, did you try with anti-inflammatory drugs like corticoid to see if you improve the efficacy and the reduction of the side effects?
Well, there are already many things to do with nucleic acid, Julien, on that. But maybe, Arnaud, you would like to comment on this, the applicability beyond nucleic acid to encapsulate other molecules? You're on mute, Arnaud.
You're on mute.
Yes, of course. The technology was developed specifically for the delivery of RNA, but we can also load because we have macroparticles, and inside of macroparticles, as Henri explained, we have also lipid nanoparticles. That it means we can load both lipophilic and hydrophilic drugs inside of macroparticles. So it means we can load a lot of kind of drugs, including, of course, steroids.
Yeah.
Steroids, yes.
Yeah. Thank you, Arnaud.
And in case your drug carrier reach the blood compartment, is it safe? That's another question from the audience.
That's a very good question. So we have already administered orally and IV without the microparticles our drug asset. And what we observed is that once administered orally, it does not go to the plasma. So, which is very good indicator for the safety. So it stays local. But even when it goes local, so we see that when administered IV, we have measured potential toxicity on the liver that we have not observed any at the moment. So meaning we stays within the normal values in terms of toxicologic effects on the liver.
Okay. And if we stick to performance, I mean, you showed us some data regarding the positive impact of your solution. What if we compare it to existing solutions that are already on the market? Do you have any comparison to that in terms
That's
of
a
performance?
good question, Julien. Can you see my screen? I think you can.
Not yet.
Oh, now you can.
Soon.
Yeah. Yeah. So we have compared to the key drug candidates being in the development, because beyond the current landscape, what is even more important for us in terms of competitor intelligence is to track the key drug assets that are in the development currently, that we know from key opinion leaders that they will be the one that will be used ultimately when we'll be on the market in the 8-10 years time frame. So we compared in the same animal model, but in non-direct studies, but in the same TNBS model, the impacts on our drug asset versus the tulisokibart from Merck and the icotrokinra from J&J. On the two key parameters, meaning the inflammation reduction as measured by the Coulomb ratio that Arnaud showed you, and on the weight gain. And what we observed is that numerically speaking, we are in the game, let's say. We are beyond them in terms of reduction of the inflammation and weight gain. Of course, this is no indirect comparison, but it means that the magnitude of the effect that we have observed is very important. Beside that, just for you to illustrate the potential of the drugs that are under clinical stage for IBD, this tulisokibart has been acquired by Merck for $10.8 billion in phase 2, so not at commercial stage, which means that when it works hard, high.
Thank you. Jan, who asked a question about the corticoid, is making an extra comment and maybe would worth an answer from you. Then steroids are far cheaper than nucleic acid. So what about your carrier plus steroid in IBD instead of why could we not make it simpler?
Because it's not only about making simpler, it's making something that's really attractive also. Yeah. And oral steroid, well, it's already available, I would say. You can improve bioavailability and something, but oral RNA, not yet on the market. So something that's very tasty for the pharma companies that will ultimately acquire us at some point. So we made the choice to develop something that's really disruptive with a strong potential. And that's what makes a difference versus the other.
Yeah. From what I know, steroid over time effect decrease also, if I'm correct. Yeah. If we go back to the different studies that you've done, which species do you plan to do with your tox studies? And do you need to consider the digestive enzymes and tract as they may be different? Are different between species like rodent versus dog versus humans. So which one may reflect the best?
The human one, and we test. Beside animal testing, we test with simulated human fluids also, which are even better predictive of the gastro-resistance in the human ultimately. Maybe Arnaud, you want to say a few words about these?
Yes, we actually to demonstrate the gastro-resistant properties of macroparticles. We perform incubation in a simulated gastric and intestinal fluid, and for gastric fluid we have enzymes. We have pepsin, and for the intestinal fluid we have also pancreatin. So we use very relevant simulated fluid recommended by the USP.
And thank you, Arnaud. And maybe to come back on your initial question, Julien, related to the species, rodent, and then for the second species, that's something that we will align with basically with the regulators. And that's why in our development, we plan to have a first early stage interaction called Interact meeting with the FDA to align with them on the second species that will be used. And there are different options, but ultimately we will have the answer after this meeting. Either a pig or an HP for different reasons, I would say.
Okay. If we go down the route, I mean, if we think a bit more about market access, we know gene therapy are often perceived as expensive. And does your solution, the microparticle and so on, does that in terms of production cost and so on, is it compatible with all the health economics that we may expect after?
That's a key point, and that's why, and that's my background, by the way, to think about these questions. And even at the beginning, you need to think about that. About ultimately, is it a viable solution? Yeah, and it has for these three reasons. So microparticles per se itself, it's not really expensive. LNP are already well-established technology, and there are huge capacity at CDMO level. Think about the COVID vaccines. There are manufacturing plants that have worked on billion level kind of units. So there are a huge capacity. And the small interfering RNA is an interesting drug modality because it's chemically produced. It's not it's not biological per se, and it's pretty cheap. It's much cheaper than mRNA. So we have planned this to be completely economically viable and attractive on the market. What will be the price? We don't know, because ultimately telling the price of a drug that will be in the market in 8 to 10 years and by marketed by someone else, we don't know. But what we have built is something that will ultimately be profitable by the pharma companies that will market it.
Okay. If we go now more to more entrepreneurial aspects, things like team and so on, for example, I mean, do you plan, I mean, regarding the staff, do you plan to hire internally at some point some drug development director, or do you plan to only work with experienced and seasoned consultants? It's one of the questions raised.
That's a good question. So we do plan. This is a recruitment plan, and we will welcome soon a chief development officer, someone with a strong background in the industry in terms of drug development and in IBD, more precisely. And so we plan to recruit gradually, and after first the closing of this pre-seed round, and also more broadly after the closing of the next round, which is planned in H2 2027, end of H1, early H2 2027. So that's a mix also of internal and external resources, which gives flexibility in terms of cost structure for the company, which is something interesting as well.
Okay. And just maybe to go back, it's probably me who missed maybe one part, or I can't recall. I mean, you said that there are plenty of applications beyond IBD. Were you talking about, application within the intestine, or more broadly with mucosa? Or things maybe.
Yeah. Good question as well. So first, our strategy focus on expanding the potential of the technology, but still in the gut. Thinking about colorectal cancer, because this is the closest application to IBD. So this is I would say what makes sense. But beside, we have potential outside this, thinking about other strategies, potentially vaccination, and other mucosa, but that's a bit early to discuss about this next phase of our potential development.
Okay, so second step will still remain in the gut, and potentially one day later, you could think about something else.
Exactly.
Okay, yeah, we have another question that are coming from the Q&A. So your product could be added on the standard of care. Do you believe a pharma would like to have an IBD franchise completing their portfolio, or to replace what they have already in their portfolio?
Well, currently, from the discussion I had with the pharma players, I refer to the 6 out of the top 10. They have different thought about that. Either as a new asset to offset, the loss of exclusivity of their drug currently on the market. For some of them, that's the strategy. For some others, that's an add-on. There are companies like namely J&J or AbbVie that are building huge portfolio. They already have several assets in IBD and they want to have more and to become leader. And there are some strong commercial synergies that they can expect from that. So that's the pillar 2. And I observe also traction from pharma companies that are not there yet. They don't have any asset in IBD and they want to have a stake of that huge market. So we could fit either one of these strategies. And beside, there are some other companies that are interested in having a technology that can deliver orally genetic medicines. That's why I speak about this dual value engine, which creates different opportunities for M&A opportunities. Yeah.
Okay, great. If we go back to those entrepreneurship question, I mean, I always like to know a bit more about people. And first question is the collaboration between you two founders. Where did it start? How did you met?
We met in Besançon in January 2025. I think over 1 year ago. We didn't celebrate, by the way. We could have, and it went fast. Honestly, I think, Arnaud, we met beginning of January. That was our first introduction through the TTO Science, which is yes, an organization in France for the evaluation of academic research. And then, Arnaud, I think we agreed together to build Oragen back in February, well, 1 year ago, basically.
Yeah. Wow. So yeah, it has been good. Yeah, it went quite fast actually. Yeah.
Yes, and in that period of time, we contracted with the TTO, securing an exclusive worldwide license on the patent. We incorporated the company. We are closing this first financial round. We have been awarded by BioEurope as one of the most promising biotech in the world. And we have been recently also awarded by one other conference, but that's the news for next days, let's say.
Okay. Impatient to know more then. Well, maybe I think we've gone through the different questions raised on the Q&A session. So maybe just a big, large question to conclude. What is your biggest operational challenge for 2026, apart from closing this round, of course?
There are challenges every day in the biotech world, honestly, Julien.
So what keeps you awake at night?
I think the biggest challenge will be for us to continue with this momentum, because momentum is in our DNA, which makes sense for a genetic medicine company. And continue with this momentum is a challenge, but I think that's that kind of challenge that we like together. So sticking with the timelines of our drug development, continue building this relationship with the pharma players. And I strongly believe that thinking right now about the M&A, the exit is very important for the development of the company, especially for you guys, investors. And we will be at BioEurope to continue these conversations with the pharma that we have initiated back in 2025. So continue building and riding on this wave of momentum is the key operational challenge, as well as all operational challenges associated with any startups, recruiting, sticking with the cash, managing the OPEX. But we are, I would say, we are used to this.
Perfect. I think it's a great conclusion. To conclude this webinar, we don't have any more questions. So a last potential word to add before we close the session.
I would like to thank you all for attending this session, for your strong interest demonstrated on Capital Cell already. I think we have close to or beyond 80% of our target. And we have discussions also with a few other investors. So we are available for any additional questions, comment, and thank you for support to make oral RNA therapy, which will be a real breakthrough in the whole industry and a strong promise for IBD patients.
Yeah, it's as you said earlier, you keep a momentum. And just as a reminder of your company's GEIR in French. So really, that means you can have a 50% discount on your tax thanks to this label. And if you have any question, you can also get in touch directly with me, and I'll be very happy to raise the question towards the management team. So my contact details, julien@capitalcell.net. Do not hesitate. And looking forward to discuss with all of you. Well, thank you very much, gentlemen.
Thank you, Julien. Thank you all.
Thank you very much.
Thank you, Arnaud. Have a good evening. Bye-bye.
Bye-bye.
Bye.